Welcome to Module 4 of Cells, Tissues and Regulation. In Topic 4.2, we will learn about the resting membrane potential. During the course of this video, open your workbooks to Module 4 and try and answer as many questions as you can from Topic 4.2. The learning objectives for this topic are to review the differences in the distribution of key ions and proteins between the intracellular and the extracellular compartments, to understand the distribution of charge on both sides of the cell membrane, and to describe the concepts of electrical disequilibrium and chemical disequilibrium as they relate to the resting membrane potential of cells. Like all cells in the body, neurons have a resting membrane potential too. However, unlike most other cells, neurons can rapidly change their membrane potential. This ability is essential for the function of neurons. The resting membrane potential, or RMP, is the potential difference in the electrical charge that is maintained across the plasma membrane. Although tiny, this charge is crucial to all the cells in the body. In neurons, it is of particular importance for the transmission of electrical signals along nerve fibers. One of the most important points to remember here is that the resting membrane potential exists only across the plasma membrane. Therefore, the plasma membrane is said to be polarized, meaning that it is not neutral but charged. However, the rest of the solutions, both inside and outside the cell, are electrically neutral. As discussed in Module 2, the extracellular and the intracellular fluid compartments have distinctive patterns of electrolytes. Extracellular fluid has a high concentration of sodium and a low concentration of potassium, whereas the intracellular fluid compartment has high concentrations of potassium and low concentrations of sodium. Furthermore, the intracellular fluid also contains anionic proteins. These negatively charged proteins are too large to diffuse across the membrane and help balance the positive charges of the intracellular cations, mainly potassium. Because of the difference in the concentration gradient, there is a constant leakage of potassium ions out of the cell and sodium ions into the cell. The sodium-potassium pump helps create and maintain the differences in the solute concentrations of sodium and potassium ions, thereby stabilizing the resting membrane potential. Because the solutes are constantly moved against their concentration gradient, energy is continuously expended. The sodium-potassium pumps consume almost 1/3 of our total caloric intake in order to maintain their function. As mentioned a moment ago, the negatively charged anionic proteins inside the cell cannot diffuse across the plasma membrane. And because of the continuous action of the sodium-potassium pump and the presence of the trapped anionic proteins, the cell is always kept in a state of chemical disequilibrium. The human body is electrically neutral. It has the same number of positive and negative charges. However, there are regions where one type of charge predominates, making the region positively or negatively charged. The plasma membrane separates the charge locally, creating an electrochemical gradient. Localized at just the membrane, there is a separation of charge. Here, the inside of the membrane is slightly more negative than the outside of the membrane. However, the rest of the solution, both inside and outside the cell, are electrically neutral. So let's review some of the basics of electrical energy. According to the law of conservation of charge, the net movement of electrical charge produced in a system is zero, which means that for every positive charge on an ion, there is an electron on another ion. This means that overall the human body is electrically neutral. Opposite charges attract, like charges repel, and energy is constantly needed to separate these charges. Insulators are materials that prevent the movement of separated charges or current through them. The plasma membrane is an excellent insulator as it separates charges locally. A conductor allows separated charges to flow freely. And finally, voltage is the measure of potential energy generated by the separation of electrical charges. Voltage can be measured in volts or millivolts. As mentioned before, the net result of active transport by the sodium-potassium pump and the containment of anionic proteins in the intracellular fluid create both a chemical and electrical gradient at the cell membrane. If given a chance, the sodium ions will move into the intracellular fluid along the concentration gradient and the electrical attraction. The positively charged sodium ions are attracted to the negatively charged anions, such as the proteins and phosphates, in the intracellular fluid. Therefore, there are two inward driving forces operating here. Because the cell membrane is both not very permeable to sodium and is also a good insulator, it prevents the inward movement of sodium and maintains the separation of charge. However, tiny amounts of sodium still leak into the cell due to the difference in these gradients, which is eventually pumped out by the sodium-potassium pump. A voltmeter is used to measure the potential difference between 2 points. When one electrode of the voltmeter is inserted into the cell and the other in the extracellular fluid, it records a voltage across the membrane of approximately -70 millivolts. The minus sign here indicates that the cytoplasmic side of the membrane is more negatively charged relative to the outside of the cell. This potential difference in the charge at the plasma membrane of a resting cell is called the resting membrane potential. The value of the resting membrane potential varies from about -40 to -90 millivolts. The resting membrane potential is the electrical gradient located at the cell membrane of each cell and is measured in volts or millivolts. So let's break down the term resting membrane potential. The term resting means that the membrane potential has reached a steady state and is not actively changing, while the term potential means the potential energy generated by the separation of charges. The electrochemical gradient created by the active transport of sodium and potassium ions across the plasma membrane is the source of stored potential energy. It is also important to realize that the trapping of ions, such as proteins and phosphates, in the intracellular fluid also plays a crucial role in maintaining and stabilizing the resting membrane potential. Potassium ions contribute the most in maintaining the resting membrane potential. The loss of potassium through the leaky channels of the plasma membrane establishes a negative membrane potential. The membrane, which is highly permeable to potassium, allows the potassium to diffuse along its concentration gradient from inside the cell into the extracellular fluid. However, the movement of potassium ions is limited because the negatively charged anions, such as proteins and phosphates that are trapped inside the cell, attract the positively charged potassium back into the cell. As the potassium leaks out into the extracellular fluid, a negative voltage or electrical gradient develops on the internal surface of the membrane. At equilibrium, there is a small net loss of potassium ions, which by itself generates a small negative charge of -90 millivolts at the internal surface of the membrane. However, because the positively charged sodium constantly leaks into the cell, it makes the resting membrane potential less negative, bringing it to around -70 millivolts. Maintaining the sodium and potassium concentrations inside the intracellular fluid is crucial for preserving the resting membrane potential. If sodium and potassium were to leak continuously in and out of the cell, both along their electrical and chemical gradients, then over time the membrane potential would be destroyed. If this were to happen, the cell could no longer conduct electrical signals. The sodium-potassium pump plays a crucial role here by transporting 3 sodium ions out for every 2 potassium ions into the cell, thereby maintaining the membrane potential. Because the sodium-potassium pump helps maintain the concentration and the electrical gradient, it is known as an electrogenic pump. From this video, we have learnt about the resting membrane potential, how the distribution of ions creates the electrical and chemical gradients, the basics of electricity, and the role of potassium ions and sodium ions in the resting membrane potential.